locally. The workflow starts here with data collection. We can input DNA sequences in two ways. First is file upload. We can directly upload FASTA files from the sequencing machines. The system automatically passes the file and extracts the DNA sequence. Second way is manual entry, where we can paste sequences directly into this text area. Notice the live base pair counter. It validates the sequence in real time, counting only valid DNA bases. All metadata is optional at the point of collection. Why? Because in field conditions, researchers might not have all environmental data immediately. They can add or update metadata later through our management system. Let me demonstrate with our sample data. This loads a real sequence from Arapaima gigas collected from Amazon basin. The results card is designed for quick, actionable insights. The sample ID at the top. This is the blockchain verified identifier. Every analysis is traceable and auditable. Then we show both the scientific name Arapaima gigas and common name giant Arapaima. The confidence matrix is crucial for decision making. These matrices help us understand not just what species was detected, but how confident we are and what conservation actions might be needed. The View Data Navigation button opens a Species Metadata DB, the central repository for all verified species records. At the top, we have a search bar where we can find records by species name, sample ID, location coordinates, or any other field in the database. Next to it, we have a status filter and action buttons, that is, adding a new record manually. Also, we can export the entire database in CSV file for further analysis. Each record has a unique blockchain verified identifier. This makes every record traceable and auditable. Then we have scientific and common names of the species, GPS coordinate, color-coded conservation status, population levels in the region, AI model confidence scores showing how certain our analysis is, edit button where researchers can update the metadata. The View Abundance Navigation button takes us to Abundance Analysis interface where we can filter the data according to the region, time frame and metric. We have instant biodiversity health indicators. Then we have comparative analysis ranked by abundance, which is incredibly valuable. This was all about our interface. When I click Start Analysis, here's what happens behind the scenes. First, using one hot encoding, each nucleotide, that is ATGC, is encoded as a binary vector. This transforms the raw DNA string into a mathematical matrix ready for analysis. The first step is blockchain registration. The sample is registered on the blockchain with a unique ID and timestamp. This creates an immutable record. The next step is dual AI analysis with hybrid model. Our CNN model analyzes the sequence patterns, providing an initial prediction with confidence scores. Simultaneously, BERTax, a transformer-based model specifically trained on taxonomic data, validates the classification. Both models run in parallel, providing independent predictions with confidence scores. As we can see, our hybrid model achieves 94.8% accuracy, significantly better than any single model. It's 10 to 20x faster than traditional BLAST while maintaining comparable accuracy. CNN excels at local pattern recognition and BERTax excels at contextual understanding. Then the system retrieves the metadata you entered. A smart contract on the blockchain is automatically triggered. This contract validates the analysis, meets quality thresholds, timestamps the analysis completion, locks the prediction to prevent tampering, and creates a cryptographic link between sample ID and results. The AI extracted features from both the models are mapped to a unified feature space. CNN extracts 512 conversion features. BERTax extracts 768 transformer embeddings. Then these are concatenated and mapped to a 256-dimensional species representation. This mapping creates a unique fingerprint for the species. Now, the prediction output is cryptographically hashed using SHA-256. The hash is stored on blockchain, creating a permanent verifiable record. Up next, we have consensus validation on output. This is our quality control step. The system checks, do CNN and BERTax agree? It must be within 5% of confidence. Does the confidence meet our threshold? It must be minimum 75%. The consensus is reached. We proceed to BLAST verification. The system queries the NCBI database through BLAST verification to cross-reference against millions of known sequences globally. This provides independent verification. Now comes the critical decision. If BLAST finds a strong match that is greater than 95% of identity, species is classified as known taxa, conservation status is pulled from IUCN Red List. Else, if the BLAST finds weak or no match that is less than 95% of identity, it is flagged as potentially new taxa and is marked as novel detection. Next step is abundance counting. Finally, the system checks if the species was detected in this region before, updates the abundance count for this location, calculates the regional density.